talking about stepping up from those lower trophic level plankton up into vertebrates so that you can head to people later in the day. So one of the things when I started ecosystem modelling about 15, 20 years ago that really amused me were these people that would talk about ecosystem models and I'd rush to the paper to read it and they'd be quite frustrated. I was a bit disappointed because I actually wanted to be at the fish end of things. So this is going to be a little bit of a discussion. A fish. This is going to be a little bit of a discussion of the many different ways that you could model invertebrates. So for those of you who don't know what this delightful animal is, it's a dugong. It's an Australian manatee, effectively. They're herbivores that go along and eat seagrass. And apparently sailors used to think they were mermaids. You have to be in the sea for a long time. I think that's a beautiful woman. So the kind of modelling I do, after that little bit of a burst on Atlantis yesterday, is actually the whole little system. What we've heard about so far is this part up here. So we've heard a bit about the physical stuff and the plankton, and now we're going to concentrate on the vertebrates. And the rest of the week is actually going to touch on the whole rest of that. So for those of you who keep telling me that you're not modelers, this is a difference equation. So basically just say the number now was the number in the past, plus any new ones that have come along, plus any that have died, minus any that have died, plus any immigration. And if you like to get a little bit fancier, you use calculus, and it's really just the same thing. The most important thing about vertebrates is, though, that there's all the different parts of the life history. And in reality, and what Eileen will show you in the next half of this talk, is that they're not really different to invertebrates. So lots of people get really fixated on vertebrates and go to enormous detail in representing them in a model. And then they'll have all the crunchy things. They'll lump them all together at the bottom. You can actually think about all the same processes that we heard about yesterday from Eileen, just for vertebrates. And a lot of the methods are the same as well. So the main parts of the life history we have to think about are how they grow. Some people think about how much metabolism is happening inside, the respiration, because that's typically where things like temperature and salinity and alkalinity pH affect the animal. So in the past, lots of people skipped over this bit, but as we get more and more worried about climate change and how it affects animals, a lot more attention is going into that. Then you've got to think about how they move, how they age. At least vertebrates don't tend to shrink. That's one twist that an invertebrate has. You can have a sea urchin this big that when it gets older is actually this big, which is a bit of a pain to have to try on. But at least the fish, it might not get any fatter, but at least it won't get any shorter. Except my grandmother ended up this tall when she started this tall, so maybe that doesn't quite work. Then you've got the mortality side of things, all the different ways that they get to die, and then there's the reproduction side of things. So if you think of a biomass tool, right? So one of the ways, for those of you who don't know what this is the Australian blobfish. It lives off Tasmania. And basically, it's really just a gelatinous mass of fish that floats about this. It's neutrally buoyant, it floats about this far off the bottom and eats anything that goes past. It's a very endangered species because when the trawlers go along, they rip it up. But it looks like a blob of biomass to me. So one of the ways of modelling, and it's used in EcoPath that we'll play with this afternoon, is not to worry about the details of how many there are or how big they are, just to say, look, if you sum all the biomass together, how much they weigh, you get this blob. And you follow how much the blob changes. So you'll have more blob added when they grow or when they have babies, and then you'll have the mortality. When they die, they disappear. The other major way of modelling animals is to actually count how many there are. So sometimes you worry about how fat or how big they are. Lots of times you just assume they're a certain size at a certain age. And then you fixate more on how they have babies and how they get new ones to swim into the system, and then how they grow. One of the most common ways of representing that kind of model is actually to start to get worried about how many little ones there are, how many medium-sized ones, how many big ones. So I don't know how many of you know much about fish, but a fish this big, a really big fish, so a fish this big, say, is about the same, produces about the same number of babies as 1,000 fish that big. So even if they're the same species, they're just a big fat one instead of a skinny small one. You really want the big fat ones because they're way better mothers. than the smaller ones. Unfortunately, fishing catches the big ones, so that we change the productivity of the stock just by fishing them, because we've changed the kind of motherhood investment they can make. So that's why people get really interested in these kind of models. So you start off with all the little babies and they grow up in size. You have mortality killing them off as they go along. Then you get to what's called a plus group. People get bored counting fish as they get older and older and older. So they just say, all right, once they're over this age, we'll just add them all together. The reason they do that is it might only be 7 years for a sardine, but when you do an orange roughy, which is a big deep water, well, not big, fish about that big, a deep water fish off Tasmania, it lives 300 years. So following that year by year is a bit of a pain, especially when I first started and we couldn't fit a matrix that big on the computer chip we were playing. So we have to think of other silly ways of doing it more. So then you have your adults up here, and they send the recruits back down to the start, and you start all over again. It's that kind of model is the most common kind of what they call stock assessment model. So every time you've got a fisheries management body in the world, whether it's inside the nation or it's one of those big international ones that cares about you, typically they're using that kind of model. So they'll have age structure, they'll follow animals through that age. They can have variable sized age, they don't have to have a smooth curve. They can have lots of spatial structure. They can have genetic stocks, not necessarily in the way that Eileen was tracking them yesterday, but they just say this area is one group that doesn't breed with this area down here, so that they can have differential pressure from a fishing boat going to the different areas. Typically what you get out the other end of the stock assessment is the kind of predicted, this is actually the spit in the current state. This is a fish from Australia, they think wasn't really fished until the late 1980s, and then they think this was roughly its biomass trajectory. So Australia's management target is to have no species less than about 48% of its unfished state. One of the problems with that we'll get to later in the week is they pretend every fish is an island, which is the opposite of what eco-park does. And they estimate that unfished state as if there were no other fish in the world. Forgetting that maybe that fish is only as productive as it is because you've already taken out all its predators. So that's one of the complications and why we move on to food-grade models. The other thing we have in Australia is a minimum stock size. So here, the management starts to tighten. If you drop below that, by the time you get to here, they close the fishery. So that's a very common method of fisheries management in most of the Western world. It's a bit harder where you can't regulate fisheries bodies. It's a little bit more chaotic, but that's kind of the basic on the theory. Now, if we get on to how you can elaborate that kind of age-structured model, you start to get a little bit more agent-based. So this is actually still differential equations, it's not agent-based yet. But instead of just assuming a growth curve, you follow the average size of the average individual. So you're still doing a lot of homogeneity, you're still smearing things and assuming that they're all kind of the same, but you're not assuming they'll follow this perfect curve. What happens is they have to find prey. So this is what we use in Atlantis. They have to find prey, it then has to fit in their mouth. So if you're an animal that eats like my son, everything fits in your mouth. But most fish, that's not actually possible. They have a gape. Anything that can't get past that gape. There's a few tricky animals that trip you up, so parasites are one. The other main one is something like the cookie cutter shark, which is a tiny little shark about that big. And it swims up to the sides of big animals, like whales, and then bites a chunk off and runs away. So they can trip you up sometimes, but most of the time you can deal with them. So once they get past both of those tests, you find the prey that they can actually eat. They eat that, and then that goes into building their body. But you split that body into two parts, just like Eileen was talking about yesterday. You have the structural part, and then in a vertebrate, that's fixed. Once they've put energy into building structure, they can never get that back. Whereas in invertebrate, some of them can reabsorb their skeletons. Then you have the fatty bit. So I ceased growing any taller when I was 11. I thought I was hugely tall at that point. Mike has since proved that I am not exactly the tallest individual in the human race. But I have tended to grow outwards over the last 20 years. And that's this part. That's the part that produces stuff for reproduction. It is the part that can be sucked back in, so to speak, if they can't find enough food to eat. So you can actually represent starvation. The products of all that kind of activity, you can have losses due to disease. You can have losses due to hitting an oxygen load that they can't escape. They can also just be eaten. All of those kind of losses can feed back into the nutrient detritus. So most of those other models that we've heard about, the biomass pools and the age-structured models before this one, they don't always have the nitrogen cycle or the nutrient cycle completed. EcoPark does, to a sense, it has a detritus pool. But Atlantis, at least when I first started writing this, was one of the few models in the world that cared about closing it because it had plankton in there from the start. That's now quite common now in end-to-end models, and there's quite a large, there's at least 15 end-to-end model types I can think of from around the world, and they've all incrementally added this part. Okay, so the basic form of the, I've just lost 3/4 of the audience, I know, but please stay with me. So the basic form of this kind of equation is the change in the vertebrates through time is dictated by how much food they find, how well they can actually assimilate it. So you don't all absorb things equally well. So, for instance, someone with my body shape seems to absorb every calorie from the smallest piece of cake possible, whereas my husband can eat the same cake and it's not going to worry him. My chief programmer can eat anything on the planet and she just doesn't change at all. So we have differential ability to assimilate, so you can represent that kind of variation, just like you can represent variation in growth. But the other part is there's different kinds of food. So I find it very much harder to get any nourishment out of a bone than I do a piece of flesh. But there's other animals that can access that bone quite readily. So you can have differential assimilation capacity based on the species and what they're actually eating. You then have the modifications based on oxygen, so you don't necessarily assimilate or metabolize as well if you're oxygen stressed. And then you've got a space limitation, and that's because there's handling and competition. So there's lots of different ways of messing each other up. You can just both race for the food at the same time, and whoever's fastest gets there first, and that's one kind of competition. Another competition is when you both get there and you have a fight about it. And some of that increases once you have space. So space limitation is quite packed. So and these are the mortality terms. So mathematically, there's lots of different ways of representing mortality in these models. Some of which are from, you see in all different kinds of models. So one of the tricks in a plankton model is to close off the top. They use a squared term. So instead of just your numbers and minus a proportion of you that die over time, you make it a quadratic instead, so it's a square term on the end. Now there's two ways you can justify that. One, your predators and prey will probably go up in step with each other. So it's not always a constant proportion if the predator's population oscillates with your own. So if there's more predators when there's lots of you, a higher proportion goes, and when you disappear, the predators disappear, it's actually a smaller proportion. So that's a good reason to have a quadratic. The other trick is it's mathematically stabilising in your model, which is really the main reason that lots of people do it, to tie off the top end of the model so everything doesn't go crazy on the top. When you're using that in an ecosystem model, you have to be careful of how heavily you use it, because what you can do is just create a lot of parallel species who are actually tied to each other. So you have to be careful about the matrix that you use. But as long as you're aware of why you've used it and then you're using it with care, those kind of things are okay. Then you have mortality due to oxygen, and predation here as well. You can have the linear mortality term as well if there's a background rate. If there's some kind of disease or genetic mutation or lethal mutations that Eileen spoke about, you can have a linear form as well. Okay, so part of the things that we worry about in Atlantis is another thing called senescence. So that's when you get to sufficiently old, you just die because you're old, not because anything's enabled you. We have age-structured age phases, so you don't have to always have one bin represent 1 year. So Eileen spoke yesterday about the fact that her bins weren't even necessarily the same length all the way through the model. So you can do it, you know, one bin is 1 year, you can do it, one bin is X number of years, so 2 years or 3 years. Or you can actually have the size of those bins shift as you go through it. And the reason that you do that is when you go sit down to write a model about a biological system, or actually any system, you don't have to treat everything at the same resolution. You think about the role that that animal has or the processes happening. Now what happens in the first year of a fish's life, it can change its diet 10 times over in that first year. And then, again when it becomes a settled animal that's swimming around, and again once it becomes an adult, so that you can chunk up the life history appropriately to capture all those changes. It's also good to have that kind of age structure in there because there's a delay in recovery. So one of the issues around biomass pools that you have to be really careful about is that because they're not following the age of the animal or the number of the animal, as soon as there's more of it, it can get bigger again. There's no delay in becoming an adult, it can just grow instantaneously. So if you put age structure in there, you have some of the real recovery delays that actually happen. There's also, you can get some of the more subtle effects of overfishing, like having a shrinking size of adult affecting reproduction. And then we can have the stuff I talked about before, where we can have gait. And the reason that one of the reasons that we follow the structure and the fat is because the amount you reproduce can be affected by your condition. So by knowing how fat they are, how good quality they are, you know what kind of food they are for a predator, you know how many babies they can have. So there's a lot of reasons to do it. Okay, the other way to do it is transition matrices. This is used still a lot on land. So if you look at the modelling of ecosystems and animals on land, it's actually not as far advanced as it is in the marine world, which seems kind of odd to go up in there and ask them, well, haven't you thought about all these other things? Because they can go out and measure it. Whereas we do modelling, particularly in Australia where we can't afford to collect data, so that we can think about what's happening in a place that we can't go on a day-to-day basis. So you can use transition matrices both for ageing and representing growth in the population, but you can also use it to represent movement between different locations. So I've played a lot already with regular grids, or at least having a grid for every space, but you could actually also represent space as just nodes. So we could have one node for that corner of the room, one node for down here, another node for over there, so we just represent them as 3 spots. And you just have a transition of how animals move between the spots. When you've got a regular grid, you can have an environment like we've been playing with the net logo, but there's lots of different ways of representing space, and so there's lots of different ways of representing movement. Some of the fancier ones are, density-dependent ones, so if you're feeling crowded, they might want to move, or if they can't find enough food, they might want to move. There's whole areas of ecology that are fixated on how to actually represent movement. Sometimes you're lucky and the fish doesn't actually want to move, so lots of reef fish, once they're on a spot, they stay there for life. The other ones have small territories, so it might be that I'm quite happy to be here. Well, if you think of my teenage daughter, she has her personal space. You can't invade her personal space or you get yelled at. You have to ask permission to give her a hug, for instance. The fish are a bit the same. They'll have their personal space, their territory, which they defend quite vigorously if something else comes inside it. One thing that's actually fairly easy though, which is a benefit. One of the other ones that's quite useful is called a random walk, where the animal just sort of randomly does wander around. But there's some tricks that you can put in that. So when they find, whether you're a shark or a little fish or a human, when you're around a good food source, you tend to tightly turn, because you want to stay on where you are. When you're about to wander off, they actually do this beautiful, and pretty much every species does it. They do a big figure of 8, just checking the local area. They can't find anything, then they go. And that's a really simple pattern that follows across pretty much every trophic level we've ever looked at. There's other aspects to movement. So you've got your day-to-day movements, your feeding ones, and then you've got long distance ones. So they might be for reproduction. Most fish don't pair up. Most fish do what they call broadcast spawn, so they just release their gametes into the water. But to increase the chances, they all go to a single spot and do it together. So they have to make a dedicated movement. There's nothing quite as sad as being on a coral reef in the Pacific and watching the last lone whale go to the spawning location and look around desperately for the females that have all been fished. The other part of it is that you can have a combination of the different methods. So, for instance, in Atlantis, in the non-spawning season, they make feeding movements. In the spawning season, they ignore the food, or some species can ignore the food and just be directed by their imperative to get to the spawning site. The other part is There's lots of different mathematical ways of representing that, so you don't have magic teleportation as if they had a TARDIS, for those of you who are Doctor Who fans of the audience, and leap from one spot to the other spot across thousands of kilometres. There's lots of maths around doing that. There's some learning. So one of the common filters that people use in ensembles to actually see if models fit and try and get them better, you can use that same kind of maths to represent learning. So animals learning what's in their environment, or humans remembering where they caught something a long time ago. The last part of movement is vertical. So animals don't just move like this, they often move like this as well. So they could be looking for food on a daily basis, or they could actually be trying to get away from predators. So lots of animals come to the surface at night to feed, because then they can't be seen by predators who would have seen them in the day. The next part of the life history is reproduction. So there's lots of ones where you're looking at the effect of reproduction without thinking about the mechanics. And that's sort of the normal ones, which is the random number. Then there's plankton-based ones where you say, okay, it's a number based on how much plankton is in the model at that location, on the argument that the larvae are themselves plankton, but they also feed on plankton. Then you have ones that stock assessment guys have been using for ages, which are actually to do with number of adults, you get roughly this number of offspring. There's a few of the more common ones there. For things like sharks or whales, you actually have a fixed number of offspring per adult, is a more typical way of doing it. You can put in some of the more unusual features, like if there's an environmental driver on that recruitment, so you can have enormous spikes. When you're producing 1 million eggs, if just a tiny % more of those get through, you can see an enormous increase in the number of recruits that turn up that year, next year. So you can have those kind of effects represented. You can have parental care, so this is a father seahorse and they brood the babies. It's not the mothers in seahorses that do it, so you can represent that kind of investment. This is an example of parental care that I saw up on Ningaloo Reef. This is a humpback whale sitting at the top of its mother's back. The reason it was doing that was because the killer whales were trying to eat it. So this mother actually had twins. She tried putting one of the twins on our boat. The killer whales had taken that. Unfortunately, I was rather glad that they had, because my boat was smaller than the whale and I could only see me going in the water with the rest of them. It took them about half an hour to get the first calf, and sadly it took them about another 6 hours to get that calf. So she lost both. But there's a humpback whale mother about every kilometre up on Ningaloo Reef that time of year. It's just everywhere. One of the interactions, thinking about the human side of things, is seeing how people interact with that. So there's a lot of tourists, they go up to see them, and then you have the head of the local Chamber of Commerce who wants to cull the whale because they're having accidents with the oil and gas ships moving through the system. So different people have different perspective on how they think about vertebrates in the system. The other way that you can represent it is you can skip over thinking about the larvae and just say, right, this many eggs are produced and that translates into this many babies at the other end, and they appear and they do everything after they've been in the plankton. That's one easy trick. Or you can actually physically, explicitly represent the larvae moving around. Now, so there's some models that only care about that part of the history, but if you're trying to close the life history, you can also use some other math tricks to represent. So this is a product called Connie that we use in Australia, whether it's larvae or an oil spill or whatever other floating thing you're worried about. You can define where they come from, and it'll give you a map of where they're likely to have turned up, how far down in the future that you're worried about. So tell you, we have a big race down the east coast of Australia every year that ends in Hobart. And if a sailor falls overboard, we actually use this software to figure out where they're going to wash up so we can try to find them. The other part of movement is you can just say, right, I give up. I don't know how they move, but I just want them to be at that location at that time of year. And that's a trick that's been used in the past just to make sure that particularly crayfish turn up in the right spot at the right time. It's quite useful, but you have to be a little bit aware when we're moving into these climate scenarios, there's quite likely the fish will shift, and using that force stuff isn't going to work. You can get into some quite complicated movements, so this is starting to get more into Eileen's individual-based area that she'll talk about in a minute. But if you do have these kind of seasonal movements, so these are actual tracks of animals and how far they've moved. So you can see that they don't tend to be homebodies, like this one's going from the north to almost the South Pole. And it's really interesting, these turtles will actually reproduce over here, come and feed over here and go back there to reproduce. So they make these enormous migrations. So if you try and include that in the model, there's a couple of things you have to be aware of. It sounds good, so you're trying to make sure everything's in the right spot at the right time to be there to be eaten or to eat something. It can make it quite a juggling exercise, as I'm sure Mike can attest. So I wonder if he's still got any hair after trying to do a blank test for the last 3 years. So there's lots of reasons to include this kind of ecology though, because it links the different parts of the system together. It's one of the coupling mechanisms in nature that you have to think about being important in the model. Okay, then we get into the really messy part of one of these kind of models. So lots of those models you can just assume they get enough to eat, and you can still get the age structure happening. But if you're going to explicitly represent how animals eat, which we'll play with a bit this afternoon in EcoSim, you have to figure out what they eat in reality. And believe me, that's one of the stinkiest jobs that you'll have. So whether you're cutting up stomachs or going through Salihoglu or whatever else you have to do to figure it out, you end up, there's some things that you can separate out quite easily and go, okay, I know what that is. Lots of times you just get this mush and you know it's some kind of fish, but you're not exactly sure what kind of fish. That's where you can use things like isotopes. So there's carbon to nitrogen ratios for particular kinds of ions can actually tell you the sort of the food chain it came from, the area it came from. So you can start to infer some of that kind of stuff. So this is an example of a really well-collected diet set from just off Alaska, where over the last 20 odd years they've been collecting all of those locations, the diet, and you can see the different diet changes through time, but also through the different ages. So not all fish eat the same thing through time, or even as they get older. Thinking about those different kind of isotopes you can use to track where animals live. So this is using isotope signatures. You can tell that where these different animals actually nested, so on the different parts of the islands. And for the animals that you might not find for a couple of years later, that's actually a really handy thing to know. You can also use it to track migration. So this is the whale, the blackhead whales up in the North Pacific. And they use the isotopes to track the fact that they're actually going to different areas to feed at different times and moving about. So that's actually been quite expensive to stick an electronic tracker on, but this is using a signature in their own. You only need a tiniest little pinprick to actually do this kind of work. It doesn't have to kill you. You can also have quite large shifts in time represented in the model. So this, for example, this is an Atlantis model in one particular box. So you sort of use grid cell like I do, it's actually use polygons in Atlantis, but in one spot this is the change in shark diet through time. So you can see it was quite variable through time. But you also get quite variable through space as well. Just like any other model, you actually have to validate it and verify it and all those kind of things. So this is a comparison to that shark diet of the observed stuff in red and the Atlantis stuff in blue. So you can see we got it roughly right. We didn't always get it right. Lobster, Atlantis thinks sharks eat a lot more lobster than the sharks think they eat. So, you know, you can always have something that doesn't quite work, but you tend to learn more when things don't work than when they do. So that's actually a good modelling tip that you should always remember. Just because your model works, you still have to go check it, because you could be right for the wrong reasons. And that's a step that most people don't. Okay, so the kinds of questions that these kinds of vertebrate models are being used to answer. Just basically how you put models together, how the real systems might work, but most of it being used to ask management questions. What would management look like? How would you sample it? What kind of rules would you use? That kind of stuff. So for instance, this is a sort of equivalent diagram to the one we saw yesterday. So each one of these square boxes is where I've used the biomass tool to represent a part of the food web. And these kind of funny shapes represent the fact that there's an age structure behind an animal. So you've got little ones on one end and big ones on the other end. So little ones on this end and big ones on the other. Okay? The reason we do that kind of stuff is we don't say where the animals are going to live. We make the rules about how they live, and then our check that the model's working is to see whether the community structure, what animals live where, that comes out of the model matches what we see in reality. So these are the different kinds of community structures you see for a big model we have in Southeast Australia. If you just zoom in on one of those, so there's about 65 groups in this model, but they don't all live everywhere. So we just zoom in the one off Eastern Bass Strait. So this is what the world probably looked like in Atlantis terms back in about 1910, before they started doing a lot of commercial fishing in that part of the world. And then by about the year 2000, it changed that much. So you can see that the small pelagic fish, the little fish, had come up a lot in size. So if we think back to my comment before about the unfished state, they think about it independent of all the other fish in the world. You get completely the wrong idea if you had this version of the world for unfished, without realising that it actually changed from that in the first place. You can also see that some of these groups have changed their structure. So we think of this one here, it's gone down to that little triangle. So what that's saying is not only have we literally lost some of the large sharks, we've lost the oldest and biggest of those large sharks. So you get a change in the shape of the age structure, and that can be an important detail to try and pick up as well. Okay, so some of the ways that we're starting to use it now is to look at human interactions with the system. So there's a lot of mathematics that makes my ears bleed behind this picture, but basically, if it's above this line, the different pressures work against each other and effectively cancel each other out. If they come out below this line, they're all working in the same direction. So they're either making the animal go up in biomass, like it's increasing, they're all working together to make it increase, or they're all working together to make it decrease. Okay, so if we think of just about temperature and acidification, so if it could play the world forward with no humans in it, but still get the level of temperature change and acidification we're expecting under climate change, around Australia it actually largely cancel each other out. You wouldn't see a huge change in the ecosystem. But you add fishing into it, and it's not just that fishing makes it worse. Fishing changes the interaction effect of acidification and temperature change. They now all act in the same direction to make the situation worse. And what's happened is effectively past fishing that we can't do anything about now. It's happened 50 years ago. If you think of an ecosystem like a melon, it went in and it gutted the melon. So the ecosystem still kind of looks like it's okay. You can see the melon has the right melon shape, it's got the rind, but there's no substance inside it. So once you hit it, it shatters. Its resilience has gone. And that's the synergistic effect of fishing. It means we have to change our fishing rules, which ironically isn't a bad thing. So if we think about Australia's action on climate change versus Australia's action on fishing, it's much easier to change the fishing rules. So we'll start to get those in place a lot faster than we learn from climate change. One of the other bigger things we can look at with something like this is the effect of fisheries-induced evolution. So we've heard about evolution and how a predator can make you want to be faster or bigger or whatever. Well, when fishing comes along, it takes the big one, so it's forcing that species to try and be smaller. So the faster that you can reproduce, you're typically smaller, the more of your genes will get through. So with this kind of fishing pressure that we've got across the bottom here for one of the most common fish species in Southeast Australia, we're actually expecting this level of evolution. So what it means is the average size of the animal is likely to have decreased by up to about 10 centimetres. Now that doesn't sound too bad, except you can think about it that even a couple of centimetre change could reduce the fecundity by as much as 50%. And if they went the whole 10 centimetres, it might drop their ability to have babies by 90%. So if you start, and when you think about the gate limitation, if they're smaller, more things can also eat them. So it has an effect on their mortality. So we played that out as well. And these are 5 of the most common fish species in Southeast Australia. This is the change in just their predation mortality, not their fishing mortality, as a result. You can see predator actually goes down because it sneaks in under the window of a predator. Being smaller actually in that particular case saved it as a juvenile. It could hide better. Most of the others got nailed. And its equivalent evolution alone was the same as fishing them twice as hard. That wasn't something that we were taking account of in the fisheries. You can see it can have quite large, from the same starting point, it can have quite large effects on the amount of animals that are there. Now I've spent a lot of time talking about what those kind of classical ODE kind of models can do. They don't necessarily do crazy ass behaviour like this very well. Yep.
For the previous one, you have jellies in there?
Jellyfish? Yes, we have jellyfish.
See the substitute fish, like think like
Jellyfish are a bit like a canary in a coal mine. Pretty much every ecosystem model in the world says if you see jellyfish go up, something's wrong. And we are seeing jellyfish go up, so something's probably wrong. So what we can't do is we can't do this kind of behaviour very well. So this is where I say, that's enough for me, and I leave it to take over. We might as well do some questions to me now while I get set up. Do you have any questions? No, usual stuff. Does that mean I talk too fast? Yeah.
So you talked about migration before, and so it's like that. The problem is that if you want to model migration, you don't really know the mechanism that drives it. But you just sort of think, okay, you know the fish should show up here, and so how do you don't really know what is it mechanism driving the migration? You just know. So how do you, if you want to have a look at some climate scenarios, how do you actually know that?
Okay, so there's some that we do know what the cause is. It's like spawning or something like that. Some of it they're moving to a good feeding location. So you can start to do that. There's other ones you can do a trick where you correlate. It's all based on correlation, so it's obviously not causation, but we use it as a starting spot. You look at the temperature there, you look at the water properties of the spots they're going to, or the environment, you know, what kind of habitat is on the bottom, what kind of animals are there, how much prey, how hot is it, that kind of stuff. And then you run the climate models forward, and you look to see where those spots will have moved. So you can do that inside an ecosystem model where you've got all the things moving around. Well, lots of people, particularly on land, have just done that full stop at a species level and said, okay, these animals are likely to shift. Someone that works at Rashid has done that for 1,500 animals, so 1,500 kinds of fish species around the world, and looked at what that means for fisheries and biodiversity potentially to the future. So you can use some mathematical tricks without having to know exactly what's driving it. But you do have to be quite conscious that can mean that if what you think is causing that correlation changes, then it might be useful. So until we learn more about it, kind of where to start. Now, if you're going to
over your career, you've built models that have spun off very simply and then become more and more and more complex to the point where you now have to have a team building these models. Where's the future?
So basically, what you see in something like Atlantis, well, actually, what you see in a lot of these FD models, if you think of it as a cupboard full of spice in a kitchen, you've got all these different kind of spices, which give you different options on how animals live. You never use all of the spices, otherwise the stew tastes disgusting. So what you have to do is be quite selective about what you pick out, and it has to be dependent on what you're trying to represent. So there will be a lot more elaboration of particular details as we learn more about the climate effects and they become key to representing what we want. But I would never recommend that you turn on all the features in Atlantis or all the features in Ecosystem or anyone else. You have to be quite selective. So a lot of the big models in the climate space have become teams as you get experts in different areas. You're starting to see that more in ecology. I've had people come up and say the years of a one-person modelling team are gone. I tend to disagree. If you feel comfortable in being a one-person modelling team and you've got the support pull which you need together, that's fine. But there is certainly an idea in science that you now have to always be in a team. But you have to be really careful. That means you don't just get a super complicated model rather than a complex model that's producing the dynamics that you're after. I use qualitative and conceptual models equally as much as they use a big model. It's kind of a step by problem.
Yeah, so I'm sort of half interested in understanding at some stage you're going to reach a time where expert opinion, people who spend all their time in the system, fishermen, biologists, whatever, start to come into your model as an active component to your model.
Yeah.
Is that where you sit?
Yeah, so when I do a modelling exercise, I sit down with pencil and paper and books at the start, and then I'll take that diagram in my back pocket with me, and then I'll go sit with the fisherman or whoever, and literally in the sand or whatever happens to be the hand, we'll draw out how they think the world works, and then I'll piece that together with my idea, or if it clashes with what science thinks, we'll discuss that through. And sometimes it's as far as you have to go. So sometimes it might just be a matrix algebra, sometimes you get into like what's called a minimum realistic, we might have a fish and a temperature or something, or you might need to go the whole way through. There's kind of no recipe book that you always must do this, always, you know, only go as complicated as you need to address the question. But you always need to talk of it to the efficient and negligent within the system. So we have a process in my modelling team called the laugh test. You make a model and you take it to an expert to see if they laugh or not. How far off am I? And sometimes the laughter is because you've completely coughed it up. But sometimes it's because they haven't thought about it. So one of the ones that I always go, yes, I got that one right, was jellyfish was a big part of the tuna diet in one of the Atlantis models for the southeast of Australia. And I went and showed it to the tuna guys and said, does this look sensible? And I was like, oh, you're right, we've never seen that in jellyfish. And I said, well, it's only happening in winter. Can I see the winter data? We've never collected it in winter. So they went out and collected it in winter and they fought jellyfish. So sometimes the models can be what looks like wrong when they're actually wrong. So you have to double check to make sure the data of the model are talking apples and apples, not apples and oranges. But it's always good to get the experts to be an expert. So stop monopolising Eileen.
and its primary prey is the Antarctic krill. So I'm just going to go through these briefly. What I want to talk about with these models is the approach that was used to set them up and the model structure. And it's really the model structure, because as Beth said, when you really get down to coding these up and looking at them, a lot of what you do with the invertebrates and the vertebrates are actually the same structures, it's just what you call them when you set them up. And then talk about the limitations of these, and then I said the three examples are Biomass, Marchipus, and then the Adelie. Alright, so the first example is from a paper by Charlie Miller that was published in Fisheries Oceanography a while ago. And what they were trying to do here is examine the Gulf of Maine, which is off the east coast of the United States, and looking at the distribution of Biomass and Marchipus in that area. And it turns out that's a very important prey item for cod and haddock, and an important part of their diet. And they're trying to figure out where the sources were, because the populations of Biomass and Marchipus over Georges Bank are not resident populations. They have to be brought in from someplace else. Right, so the thought was, well, they came up here off of Canada, coming out here in the Gulf of Maine. All right, so that's sort of the setup. And Beth just said you set these models up within a context of questions that you want to answer. And that was the answer that, or that was the question that they were looking at here. So they had some various places in here where they thought the zooplankton might be coming from, and that's what they wanted to test. All right, now the individual-based model is structured around the life history of the animal. And I think as Beth mentioned before, you have to understand the animal or the organism that you're trying to model here. And here the animal's life history is providing the framework. All right, there's an egg, these various juvenile copepidid stages. This animal goes into diapause, which is a resting stage, part of its life history. And when it does that, it goes deep in the water, it hangs out during the winter, comes back up as it turns into an adult, and then the adults spawn in the surface waters. All right, so this life history defined the structure of the model, and each of these stages has different attributes, different characteristics, different behaviors, biology, that kind of thing, and they use the available data to parameterize these different stages and the different attributes of the stages, and then when you do that, you end up having to track those attributes in space and time. All right, so they set up this, as I said, an individual-based model to look at this and go through these different stages. All right, now, as I said, there are attributes, and what's shown here are the vectors, if you will, or the attributes of each of the stages. I just picked a few of them. This is the diapause stage, the resting stage, and these are what are tracked for these animals, okay, whether it's alive or dead, whether it's inactive or ready to molt. Right? And that's easy to say, but when you try to write the equations or the algorithms to decide whether the animal is going to be still in diapause, what cues it to come out of diapause, and then if it molts. There's a lot of tracking where it is in latitude, longitude, and depth. And that's important because in this model, the cue coming out of diapause is assumed to be the change in the light as you go from winter into spring. All right, so that's the resting vector. The female part of the model, all right, you have to know the age since the female matured so that she's ready to produce eggs. All right, whether or not her clutch or her eggs are ready to be released, and then knowing where it is in space in X, Y, and Z coordinates. All right, and then again, the age since maturation seems easy to say, but you have to find a way to calculate how, all right, this animal became an adult here, now she's maturing, and now she's ready to spawn. Right, and and then the nuclei here, right, going through what stage it's in, whether or not it's ready to molt between stages, and again, sorting all that out. But these are the variables that have to be tracked for every individual in this model, right? And so each of these is tracked and kept up with through the calculation. So these kinds of models tend to be computationally intense because of this, and this is just and this was not a particularly difficult one. And one of the criticisms I hear a lot of times with these individual-based models is, well, so what, you can't really verify it, you can't really do anything with it, but you really can't because you have these kinds of data here, all right, where you verify the population structure and economy. Right? You might not be able to get absolute numbers, but you would certainly know that the eggs from observations, and here you use sort of a whole range of kinds of things. You know, the egg should appear at a particular time because you see the different stages, and this is based upon observational data. And so you can, in fact, verify this kind of model through using this kind of information about when things should be appearing, the time period it takes to go from a nuclei into a copipidid or a juvenile into an adult. There are ways to look at the verification of these and know whether or not those algorithms you put for each of these components of the model are in fact producing the right kind of results. All right, so this is one way to do it. This is in time, the number of days from the solstice, which is what they're using in this particular model, is the cue for the animals to come out of diapause, and going over this period when you get to reproductive events. And the other way to do it is this is a spatial distribution, and because this is all time, they also have to have a circulation model to move these particles around, and this is a spatial distribution of the life history stages, all right, where the yellow ones are the adults, the blue ones are the eggs and octi, and the copipidins and things. And you can see this is on one day of day 160 and day 190, 30 days later. And you can verify, again, the model by the distribution of where these animal stages are in this particular area. All right, so this is again, computationally intense way to do it, these models tend to be computationally intense, as I said, because this is embedded in a circulation model. All right, the limitations in this particular model, all right, is that the growth and the reproduction of this animal was assumed to be controlled only by temperature. And that works for a lot of things, but we also know that things like food quality and food quantity are important to the growth. And in this particular model, that wasn't included. And Beth mentioned things about knowing the diet, which I'll come to here in a minute. It assumed that light control emerges from diapause. We don't know really what controls things like that. And other animals have different behaviors, in terms of activity that are related to seasonal cycles. And in this particular case, there's not any feedback between the zooplankton and its environment. It's not extracting food from the environment or giving food back. All right, so what Beth had just showed is a way in which you interact the individual-based kinds of models, which you tend to have for the higher trophic levels with the environment. And this is a second paper in the same volume stage has its own needs and its own attributes, and how it accesses this rest of the ecosystem is dependent upon what those are. All right, so if you take that and roll it up, now the model looks like this, where there is this ecosystem model over here, or lower trophic level model, more correctly, with the phytoplankton, microzooplankton, and detritus. There's all this detail over here for the individuals, for the particles. And in this case, the individual particles are actually combined to form a population because it's using what's called a Lagrangian ensemble particle, where each particle is a whole group of individuals with some variability associated with it. Right, the important thing in this one is that there are multiple food sources, multiple diet, or different diet structure for the different stages of this particular zooplankton, and also tracking the lipid. All right, so this is an example of you don't just eat the food, you're needing a particular type of thing from the food, in this case lipid, because that's what's used to build reproductive tissue, and then also to do the female needs that for eggs. All right, so this is a food quality issue as well as the food quantity in this particular model. All right, and the connections between this structured model over here for the zooplankton and the environment are through ingestion, through egestion, and also through mortality. Right? And that's going to be the same, even though this is a zooplankton, that's going to be the same for just about any animal. You know, it has to eat, it has waste products that go back into the environment through detritus and nutrients, and then the mortality ends up going back into the environment, into the lower trophic levels in this case. So these are if you have one of these models, these are where you have to have the connections that go into these larger models. And the Atlantis model that Beth was just showing has these same kinds of connections between each of those members of the higher trophic level and the lower trophic level environment, which means these are where you need to put some of your information, or this is where you need to put your effort in being able to define the connections in this model. Okay, model setup. All right, again, it's these vectors of looking at the attributes of particles. All right, this is just one of them. It's based on the life history stage. It's looking at the mortality of individuals in the particle. This paper has a huge table in it where it gives all the attributes of the particles. The reason I put this up here is not because I expect you to really look at it, but if you look at this, there are not any equations here. It's if this, then that, if this, then go there. All right, so once you get to this level of detail in these models, you don't have an algorithm. You're doing testing all the time that if it's bigger than this, then you go there. If it's smaller than this, then go here. All right, and that's an issue with these kinds of models because that kind of structure doesn't work well on parallel machines. All right, and so they're not algorithms in the sense that we have governing equations or some type of equation that's easily applied across all of this, because it's all different when you go through your testing with different situations. Right, and that's particularly when you start adding in things like lipid, carbohydrate, and protein in terms of food quality. You want this much carbohydrate, but it has to be in the ratio that's appropriate for the lipid and the protein. All right, so there's a lot of testing that goes on to do that. All right, the results of this model, you can do things like, this is the phytoplankton concentration, and because there is a link directly between the zooplankton, each of the stages, and the phytoplankton, you can in fact estimate grazing control on phytoplankton blooms. And you could do that in the aggregate kind of models that we heard about yesterday, but in this case you can say it's the adults doing it, not the smaller ones, or vice versa, and you can apportion what is actually controlling the distribution of this phytoplankton bloom. You can also look at the characteristics of individual particles, which is shown here, estimating the growth, where it is in time, its migration. migration, looking at its reproduction, and the survival. Right? So this gives you 4 examples of the particles that were analyzed to look at those characteristics. And then you can take the Lagrangian ensemble particles and turn it back into a biomass. And here this is the distribution of the zooplankton biomass in this model. So when you start combining the Lagrangian and the Eulerian, which is what the words are. The Lagrangian is that you track something in space and time. Eulerian is you sit at a point and look at what's going on. So in this case, what this has done is combine those 2 approaches to modeling, and you get a larger variety or a better look at what's going on. Right, but the limitations are the food quantity and quality. This particular test shows that those are really important, but the focus here is only on lipid. There's also carbohydrate and protein that are really important for somatic growth. Right, and in this particular model, these were assumed to just be there in the quantities that were needed. And one of the things has to happen when a lot of the models that are doing this now are not just using chlorophyll food or whatever, they're actually breaking chlorophyll down into the biochemical constituents. Once you start doing that, then you have to always check ratios because you have to make sure that they stay in the right ratios for the tissue growth in the particular organism. Right, so that makes it a little more difficult. But it also gives you a more mechanistic basis for understanding what was controlling growth and reproduction. All right, the limitation here of numbers via the Lagrangian ensemble particles. This is a way to get around the computation that's required when you have millions. You want to have millions of particles. All right, in this particular model, there was a limited role of the physical environment of the habitat because it's a one-dimensional model, and it's also limited in terms of the analysis of variability because of that. Okay. So, but the point to make here is that this is a good example of how to put a lot of complexity about an organism in a model, in this individual-based model, take that and expand it to a population, but also allow that to be interfaced with a lower trophic level model. Okay, now, Beth put up this particular figure here. I'm going to talk about an Adelie penguin model that actually Marsh did early on. Now, I realize this is not an Adelie penguin, it is an emperor penguin, but I couldn't find the equivalent picture or diagram for an Adelie. All right, and in this case, the Adelies come on land in late October. They build their nest out of rocks along the western Antarctic peninsulas where this was applied, and then they lay their eggs, right? And unlike the emperors here, they actually lay their eggs in a rock nest, and then they're usually 2 eggs per breeding pair, right? The eggs stay there for a while, they hatch, and then the parents take turns going and feeding the chick. One parent stays at the nest while the chick is small. The parent goes out to sea, feeds, brings the food back, feeds the chick. They take turns doing that until the chicks get large enough that both parents need to be going to sea to feed it, and then they go into what's called a creche, where all the chicks hang out together, and then eventually the parents say, okay, I'm done, you're out of here, and then the chicks fledge, right? And one of the ways the parents get the chicks to fledge is they just quit feeding them. I thought about doing that with my son and see if I can get him to do it out of the house, but it hasn't worked yet. But anyway, the chicks then leave and they go out to sea. All right, so what Barish did was to look at an individual based model that goes from this egg all the way up here to the point where the chicks fledge. All right, now, why was this of interest? Okay. One of the things that, one of the reasons this came about is that, okay, I'm not a penguin biologist, neither is Marsh, but we work with people who are, and in fact, someone who had been working with the Adelie penguin along the Western Antarctic Peninsula for about 30 years said, you know, I've noticed that in spite of what goes on in the environment, when the chicks fledge, their weight is always right in here and about 3 kilograms. And so wondering why it might be that that was always the fledge weight of the chick. And they don't have a lot of tolerance. If they're a little bit less than that, the chicks don't survive. If they're a little bit more, they actually don't survive as well either. So he was asking the question, why would this be so constant? All right, and so we put together a little model, or Marge put together a little model to look at how that could be. All right, so there are a lot of things that could happen when the chicks can modify their energetic or metabolic needs to get this optimal biomass that independent of what goes on, or the parent could modify the way it brought the food to the chick in terms of the timing and the frequency of the food to compensate for variability in the environment. So low food conditions or whatever, the parent just brings more, or high food, it doesn't have to bring as much. So anyway, so there were some suggestions, and this came from conversations with the people looking at this particular penguin. And I think maybe that's the message here, is you don't do it all yourself. You work with the people that know or understand the animal, understand its biology or its ecology. And what Beth just said, you take their model, you show it to them, and say, does this make sense? And then you iterate on that until you get something that everybody agrees on, and then you go ahead and try to do it. All right, so, so first thing here is to think about what do you feed to the chick? All right, well, Bill Fraser, who's the person we were working with, had looked at stomach contents of the adult penguin coming back to feed the chick. And these are some data from 1988-89. This is from a site along the West Antarctic Peninsula. This particular penguin eats primarily krill, and that's probably what it feeds its chick. So what he showed here is the krill length distribution or frequency distribution that was in the stomach of the parents returning to feed the chicks. All right, Antarctic krill, adult krill, maximum size is up around about maybe 60 millimeters. All right, so what he showed here is that in this particular year, the chicks were being fed these large krill in here, and in this year, they were being fed this smaller krill over here. All right, so you say, all right, but then you start looking at how much energy is in the food. All right, the larger krill have a lot more energy because these are actually the gravid females. So you take, fewer of these give you more energy than these, which are down here, which are the lower energy food. So the quality of this particular food varies pretty much between these two distributions. All right, so how do you get, there we go. All right, so how do you get a penguin to give you a diet sample? All right, well, you might not recognize this person right here, but this is Barsh. And this is a little Adelie penguin that he's babysitting while it's being, waiting its turn to have its stomach sample taken. And so in this case, with the Adelies, if you look here, there's a little Adelie right here. And this person has a carboy of warm salt water, and she's slowly pumping it through this tube into the Adelie's stomach, and the tube's been put down in the stomach. So this is another student here is holding that. So she's pumping this nice warm salt water down into the penguin's stomach, and then the stomach gets full of that warm water, and the penguin sort of goes like that. And so you turn it over and you gently squeeze it, and it throws up in this bucket. Right, so this is a real high-tech operation. Right, so you got the penguin throw up in the bucket here, and so you look at that, and this is what was in the penguin's stomach. Right, this is Antarctic krill over here, and so you take all these and you measure them, and you look at the sex of them and all that, and you do all kinds of things with this. But the point I want to make, which I'll come back to in a moment, this is what was in the stomach for krill. There's also all of this, which is fish. Right, and I'll come back to the fact that there the differences in diet here in a minute. But so when you do these kinds of things, and somebody says we need a diet sample, well, somebody has to go out and get it. And for the vertebrates, it's this kind of thing. You pump stomachs, you look at their scat, you know, you do things like that that are, it's probably why I got out of biology, but anyway, you get that kind of information, all right, and then you have to set up, the model. You want to relate age in this case, because we don't know, you know, the chicks, you don't follow the same chick every day. You have to be able to somehow say what age the chick is. All right. So one way, a non-invasive way to do it is to get to measure their colon, which is their beak. All right, and that's what they're doing here. This is an adult Adelie, but with the chicks, they just grab them, weigh them, and measure their beak, and then they can you can do a regression on it to get at the age. And as Beth mentioned, you need age structure. And that's not an easy thing to do because you don't follow the same individual all the time. And that's why you have to infer it from things like age length diagrams, age weight diagrams. It's not an easy thing to put into these models, although it sounds easy to say you have an age-based model. It makes sense where everything grows up and gets older. All right, so this is a regression for relating beak length or colon length to penguin age. All right, and this is the model that was used, a very simple model for getting the change in chick mass over time. It's just assimilation. This is the respiration from when the chicks are active, like running around the colony, and this is when they're sitting and resting, particularly in the crash period when the adults are not there. They're just sort of hanging out with each other trying to stay warm. All right, so again, a deceptively simple looking equation, but there's a lot of biology and a lot of understanding that has to go behind this. All right, so the results here, this is age, chick age over time, chick age versus chick weight or mass here, and for 2 time periods. All right, now this period right here is the egg. This is eggs respire, so there is a loss. I'm sorry, no. This is when the hatching, excuse me, is right here. The egg part was back here, and this starts when the egg hatches, and then the chick begins to grow. All right, so what happens here is is that right? So it's growing here. Yeah. And what the line is the simulation, and these dots are the observations for these 2 years. All right, so you look at this, well, the model is sort of doing okay. The chick is growing here. The drop in mass right here is when the parents quit feeding the chick, all right, to make it go fledge. All right, and at this point, the chick takes the chick about 3 days to realize mom and dad aren't coming back, and I need to sort of get out of the house and do this on my own. And that's when they go into the ocean at that point and start trying to learn to swim and go find their food. All right, so most of these fledge weights are about 3 kilograms, which is right, but when you start looking at the differences, like in this case here, we're looking at it and say, oh, the model's doing all right, but it really isn't because it's underestimated the growth of these chicks, and here as well, and here it's sort of underestimating and overestimating. And as Beth said, that's when you start learning things, and that's when you start trying to figure out what's actually going on. All right, well, what Barish did here was to do a series of simulations changing chick assimilation efficiency, changing respiration rates, and seeing where that moved the simulation relative to the observations. All right, and in some of these it helped, but in others it really didn't. And so in the paper there is a discussion of what's going on here and why that might be. All right. Only one prey item, and here's some of the ideas, the limitations here is there was only one prey item, which is the krill. And we know that the adults can bring back fish as well. And subsequent work went and looked at that, and it turned out that there is a mixture of fish and krill in the diet. And when there are years of bad krill availability, the adults tend to bring back more fish. And it turns out the fish are energetically more favorable to the growth of the chicks, so giving them a few more fish can in fact make up for the difference that they're not getting from the anaerobic krill. So the important thing here, and as Beth had said, diet changes over time, and that's an important connection between the ingestion part and the environment in the urine. The other thing that's not in this model is thermal regulation. All right, that the animals are sitting out on, the chicks are sitting out on the land. One one of the things that's happening along the West Antarctic Peninsula now with climate warming is that it's starting to rain during the summer. The chicks get wet, and it really screws up their thermal regulation, even when they are in depressions, and so they lose a lot of energy as a result of just trying to stay warm or dealing with wet feathers. All right, so thermal regulation turns out to be important, and the trade-offs in the physiology, like decreasing respiration rate, increasing assimilation, those were all imposed. They were done in an ad hoc way and not based upon any real observations. So that's an area that when you start doing these kinds of models, you really need to talk to people who are physiologists and let them help you figure out how to do this. The other thing is the role of the habitat, which was imposed in this model through the diet that the animal was given. All right, that the adult was bringing to the chick. All right, and again, what Beth showed a little while ago is the shark diet changed over time. That's a representation of what's going on in the environment, and the environment in this model is imposed through what you impose from the diet. So if that's the approach to taking these models, it means that you really have to know the connection between what the animal is eating and the environment here. All right, if you want to project climate change effects, you really need to do the life history, the physiology, the ecology, the interaction with the habitat, and if you want to add fishing on top of all of that, it's a lot of information to pull together in order to do one of these models, which is why we tend to work with groups of people, not necessarily people who are modelers, but the people making the observations who actually understand the bigger picture, which in the case of the penguin model, the penguin biologist we're working with is that type of person where he would be able to say, no, your solutions look stupid, that's not how a penguin grows, and then you can go back and revise the model to go with that. All right, last couple of comments here is that I put this up yesterday where this is the structure for a particle that is at the mercy of the flow, a planktonic particle with all this circulation and everything. But you can take this kind of model and then just put the vertebrate part up here where it's accessing what's going on through all of this in here. And which is what Beth was showing, we'll talk about later today. And as an example of this is a figure from Dan Costa, and I think Luis has worked with a lot of these data techniques. You did this, I don't know. But this is 3 seal species along the West Antarctic Peninsula that were tagged. And you can see this is elephant seal, the crab-eater seal, and the Waddell seal. The thing that comes out of this is that each of these seals is using a different part of the continental shelf. Right? And because of that, they're accessing different food environments, different physical environments. And so if you were to try to do a model with these seals in it for this area and looking at prey availability and whatever, it matters where they are on the shelf in terms of what they're accessing and what they are eating. And the last comment here, the previous slide was from Dan Costa, who's Luis's major professor at University of California, Santa Cruz. But the other comment I'll make from some of the presentations we've had and some of the comments is that these large animals, like seals, whales, birds or whatever, this is a series of papers. Again, this is from Dan Costa here, indicating that these top predators actually are being recognized as being important in phytogenetic cycles. Right? And so there's a connection that goes on between these. Right? As an example, krill eat phytoplankton, and they release the iron from the phytoplankton when they feed. And we heard yesterday about iron limitation. We've heard that several times. Right? For phytoplankton, so the krill eat phytoplankton. They feed the sediments. So they're moving up and down. The Antarctic krill are accessing phytoplankton and the iron and the nutrients in it. And there's several papers that have come out right now about whales eating the krill that live in the Southern Ocean where it is iron limited. They come back up to the surface where they defecate, their fecal matter goes back into the surface waters, and it puts iron back into the surface where it stimulates phytoplankton blooms. So there are a lot of papers now. The most recent one that came out is called Baleen Whales as Farmers of the Sea. And one of the suggestions for the low productivity in the Southern Ocean is the removal of the baleen whales through whaling. All right, and there's a lot of interest now in going back and recalculating the productivity in the Southern Ocean for the time when the baleen whales were there. So there's apparently a direct connection between these large animals, at least in the Southern Ocean, and something like iron cycling and counter reproduction. So they have also an effect on the ocean carbon budget. Some of these papers here are doing calculations of the amount of carbon that's found under these animals and then how it gets back into the system. All right, and sperm whales returns a lot of the significant amounts of carbon and iron. As an example, these animals feed deep. They go down. They're effective at moving things from deep layers back to the surface ocean. And the last part here is it says that the vertically migrating animals have access to what's going on deeper, which then they come back to the sea surface and release. Okay. In here, the model frameworks I've been talking about apply across a range of species, a range of environments. But I think something that is needed and something to think about is extending these individual-based approaches to the population and allowing for the genetic variability in the population. The habitat models are becoming important, or not becoming important, but are important in terms of how the animal uses the habitat, because it's not all done the same way. And through foraging models, because these animals do that, like the penguin chick, we impose the foraging behavior of parents by altering the timing of when the adults arrive to feed the chick. Right? That assumed a whole lot about what was going on as the parents went out to sea and came back, rather than, that's particularly important for these large vertebrate species, is their foraging approaches. And then approaches for linking the food web and the biogeochemical models. This end-to-end idea is part of why we're here, and it's also a very active area of research. So I'll stop at that point and ask you if there are any questions. Thanks.
So if fish is a better source of nutrition for the penguins, why krill?
Because their krill is more accessible most of the time, and the other thing that's happened, which I haven't gone into here, is that the fish species that was in that penguin stomach is actually going extinct along the West Antarctic Peninsula. It's Chlorograma Antarctic silverfish. And it needs sea ice for its eggs as a nursery ground, and the sea ice is disappearing along the West Antarctic Peninsula. So a concern is now what's going to happen with the Adelie penguin now that that food source is gone. And that's an animal that seems to be pretty hardwired for particular prey species. And in fact, the Adelie populations along the Antarctic Peninsula are severely declining, and some of the colonies have already gone extinct. So
That's part of Jose Torres' work, right?
yeah, Jose Torres has done that, and Bill Frazier has also been looking at stomach contents, and you can see a major change from a diet that goes from, say, half fish to half krill to a diet that's almost half krill along the northern part of the peninsula.
I was just wondering if you keep the timing after this process. If you keep the timing after this process, no?
Oh, no, the penguin loses its meal. They get really, yeah, and the people who do the diet sampling, they try to make sure they don't sample the same penguin more than once, because they lose the meal.
What if you better than nine?
Yeah, it's a non-lethal way of obtaining a sample.
It's an annoying one.
What is the ratio between krill and fish nowadays in terms of available
very small part of the plan.
Other fish, I'm going to replace.
No, and that's, there are other fish, Electrona and Arcticum is, the Electrona is coming into that area, but it stays deeper, and Adelie penguins are visual feeders. They pick out each individual prey, and so the Electrona is deeper. But the other thing, Adelies are, as Beth mentioned, they're gate limited, right? And so they can't really use that resource. So it's kind of like having, being in a desert and the only water you have to drink is salt water. It's the equivalent thing for them. Yes.
In the Mediterranean, we are investigating conch seals, and rather than making them throwing out to inspect their diet, we examine their feces. Could it be possible also for this case, rather than making them throwing out, would it be better if you investigate feces and collect hard particles like otoliths or this kind of thing?
Well, krill don't have the hard parts, and it's very hard to measure what comes. You want to know the length frequency
Next frequency in the diet.
in the diet. All right, and you don't get that from the feces. I think Louise has done a lot of that.
Yeah.
You can get otoliths.
You can only get out of
Yeah, you get krill eyes. Yeah, and that was a thing for a while. People were trying to take the eyes from the krill and use that as a way to estimate the length and things, and it just doesn't work.
So Jacobo talked about how the agent-based models have the potential to address complex adaptive systems and emerging behaviors. And I was wondering if the IBM models also have showing signs of emerging phenomena in that sense.
So individual-based models and agent-based models are effectively the same.
Yeah, they're the same.
So what's the difference between an agent and an individual?
So it comes down to a little bit of the history of the model. When these models started, they were mainly on people, they really were individuals. But as the method has grown, they've realised that you can use even other kinds of models. So that's why it became an agent. So when I talk about an agent-based model, my sharks or my penguins will be individuals. But as I slide down through the trophic web, my plankton will be an NPZD. So it's an agent in the system. My environment, my hydrodynamics model will be an agent. So they all have interactions that go with each other, but it's not really an individual wave or an individual plankton that I'm following. So yeah, that's why it's just a jargon terminology.
So in that regard, if you connect up all these agent-based models, then I would hope that you could get some emergent behavior.
In game theory, we use agent as a flag.
Right.
So as a flag, and the flag can be anything from an individual to a nation to a group of people. We are an agent here, this class as a whole.